Hello and welcome to Chapter 15. This is Part 1 of Regulation of Gene Expression. And so we've learned about gene expression, how genes are transcribed and translated. And this is really about what are the signals that say this gene gets turned on or this gene gets turned off, or how much this gene gets turned on and when that happens. This is all very important for how an organism functions. So we can first think about what happens in eukaryotes versus prokaryotes. Now, both kinds of organisms are maintaining homeostasis. Prokaryotes do this a little less rigidly than eukaryotes do. Both are responding to environmental signals. But if you think about a typical prokaryote as a single-celled organism, and that doesn't mean they can't aggregate together, but generally a single-celled organism, there's a much more direct connection with that environment. Like, I'm a cell, here's my cell wall, and boom, out there is the environment. Whereas eukaryotic organisms, particularly multicellular organisms, have specialized cells. Those cells carry out a certain function, and other kinds of cells are doing some other sort of function, and those are interacting together in that larger multicellular organism. So let's think about that with a question. So altering patterns of gene expression in prokaryotes would most likely contribute to the organism's survival in which of these following ways? All right, hopefully that's an easy one. This idea that they can respond to changing environmental conditions. A new food source is available, turn on these genes that are important for processing that food source, turn off these other ones that aren't needed at this time. Another place that gene expression is really important, and here we're coming back to eukaryotes and multicellular organisms, is controlling development. In the next chapter, this is an area of focus, but it's important to mention here and think about what's happening. You know, say humans, we start as a single-celled organism, and then 2 cells and 4 cells, etc. And then those cells differentiate and know sort of what's the left of this organism, what's the head, what's the tail, and so on. And so it's helpful to remember back to our central dogma of molecular biology, this flow of information, DNA to RNA to protein through transcription and translation, and think about where can we regulate this expression of genes. And so if we look at any of these stages, and as we'll see, we can regulate expression at any one of these points. And this brings up another concept, this idea of cost, where if we're regulating, say, at the DNA level before a gene is even transcribed, that's potentially less energetically costly versus, say, producing an entire protein at the very end and then just degrading it. Oh, we don't need that anymore. There's been a huge investment to get to that point of having that protein. And so it can be helpful to think about this in terms of, you know, a cost and energetic cost and where we regulate transcription. So I can write this energy. We're talking about the energy cost here. And we can start at the DNA level, RNA, protein, and then of course we have transcription and translation that's happening that get between those transitions. And if we just think about this, again, about cost, it's going to cost more to regulate at the protein level because we've invested in building that protein versus, say, regulating at the RNA level or, as we'll see, even regulating at the DNA level. And sometimes it's not sort of a matter of a choice of energy cost, of course. The other important point to recognize here is gene expression can be regulated at all these levels. So, you know, say a protein is produced, maybe it might cause a problem in a certain situation and we don't need that anymore. It can be broken down or deactivated, or the gene expression can be shut off at the RNA level and so on. And so that's important to remember. And as we'll see, this can work at these many different levels. And that's really driven home when we look at eukaryotic gene expression. We'll look at prokaryotes too. But if we look at what's happening in eukaryotic gene expression, We can see chromatin modification, and that can turn genes on and off. Transcriptional modification, RNA can be processed. RNA can be blocked and degraded. Whether or not a polypeptide is produced correctly by the ribosome and goes where it needs to go can be influenced. The protein can be destroyed. The protein can be switched off, and so on and so forth. Let's think first, though, about controlling transcription. If we think historically, this was sort of the first place when we're looking at regulating gene expression where we started to learn about this. And it makes sense, right? Here's a gene in the genome and DNA. Does that gene get turned on? Well, it has to do with binding of proteins that are called, say, transcription factors or other regulatory proteins. And these might block the transcription of a gene. They might act as a place, sort of a signal, like I'm bound to this gene, come over here, let's turn this thing on, let's make some transcripts. And so these are regulatory proteins. And we can see just sort of the general shape of one of these regulatory proteins often fits in size-wise here with this double helical strand. Now, if we think about what's happening in either prokaryotes or eukaryotes, and think about metabolic pathways, there are often multiple steps in metabolic pathways. We talked about how that's important for regulating. And here's where we're really seeing that happen. So in this case here, we have a tryptophan metabolic pathway, so converting some precursors through multiple steps into the amino acid tryptophan. And tryptophan, if you've got enough of it, will switch off the expression of these proteins. So here you see this negative regulation. So, hey, we got enough tryptophan, we don't need any more. The end product actually acts as a negative feedback on an earlier step. And so we can think about regulation as both either negative or positive. Here's a general negative control example. Here we have, in the case, no negative control happening. RNA polymerase zips along and transcribes this gene. But it's when the regulatory protein is present. So here's the negative control. It is blocking, in this case, RNA polymerase. So boom, this isn't going to happen. The alternative would be positive control. So here's a different example. Well, RNA polymerase is bound to this gene, but it's not working. It needs something else. And so you need this other regulatory protein, as we see here, and that will trigger the transcription. So actually make that happen. So that's positive control. Sometimes I like to think about this like driving a car. The negative control are like the brakes, on a hill. The car is going to roll down that hill, and to make it stop, you have to put on the brakes. So that's the negative control. The other way around here, the positive control, your car is just sitting on a flat surface. It's not rolling anywhere until you give it some gas, and that's the positive control. So let's look at some examples of this with prokaryotes. Now, with prokaryotic genes, they're often arranged in groups of connected or related genes into something called an operon. And so what we see with an operon here is, in this case, we have these different genes, X, Y, and Z. Instead of them being separate with each their own promoter, you have 3 in a row right after a single promoter here. So if you turn this on, basically, you're going to produce these 3 different gene products. Something else that you'll see here is this operator region. And it acts as a regulatory switch, either turning this on or off, again, as we'll see in some examples here. And that's usually within this control region or promoter region. And then together, this all forms the operon, the control region and the actual genes. And so in prokaryotes, operons can either be induced when they're needed. So there's that example where some metabolic pathway that you don't need right now, it's just not on. It's not running at all. It's just sitting there quietly. None of the genes are being expressed until that substrate comes along, and then, okay, let's switch these genes on. So that's induction. The alternative idea is some operons are repressed. So the metabolic pathway isn't needed, but it won't just sit there. It would be expressed otherwise, so it has to actually be repressed. Something has to block it. There's that negative control. And so we'll start with an example of that. This is called the trip operon. And so this produces gene products that are important for tryptophan synthesis. So that's an amino acid. So when a bacterium, this is all work from E. coli. So E. coli runs out of tryptophan, it can synthesize its own. It can take precursors and using these enzymes make tryptophan. And so when tryptophan is low, it needs these genes. And so what's happening here normally, say tryptophan's low, RNA polymerase binds to the promoter, goes through the operator, transcribes these genes, everything is great, make a whole bunch of tryptophan, wonderful. There's another gene that's related to this, though. It's got its own promoter here, so this is not part of the same operon. This is a regulatory gene that's separate but near this. And this produces this repressor protein. And notice it's inactive right now. So we need tryptophan. Our trip operon is being produced here, transcribed, great, wonderful. But at some point, maybe we end up with plenty of tryptophan. You don't need it anymore. Well, let's stop making these enzymes because we have a ton of tryptophan. You just don't need it. Or there's tryptophan in the culture media or the environment that the bacterium is in. Doesn't need it anymore. So what happens is this. Tryptophan will bind to this repressor protein that's just been sitting inactive. So when there's enough tryptophan, they'll start to stick to this. And this is called a co-repressor in this example. And here they are together forming the active repressor. And you can see the shape here and where this is probably going. It will bind to the operator region. And now this blocks transcription. So when there's lots of tryptophan, we don't need to express this tryptophan synthesizing operon. And therefore, this active repressor will now block that transcription. All right, so the trip operon is repressible. Which of the following correctly describes this operon? All right, hopefully that was pretty easy. Yeah, if you were to add tryptophan to, say, our culture media, bacterium doesn't need it anymore, then the trip operon would be switched off. All right, so let's look at a different kind of negative control. This is still negative control, but we'll see how this is different from the trip operon. And so this example uses the lac operon. And so the lac operon has to do with lactose metabolism in E. coli and other prokaryotes. And as we'll see here, we can have a quick look now. We'll see this again. We have this regulatory region. So here's this operator. There's a promoter for the lac operon. And then there's several genes here that are important for bringing lactose into the cell and splitting it. Lactose is a disaccharide. There's also this other gene, this regulatory gene, like we saw with the trip operon. So it's just a little bit upstream. It's got its own promoter, and it produces this repressor. Okay, so the lac operon is inducible. It's still controlled in a negative way. So normally it is off. And it's negatively regulated by a repressor protein. And so the repressor will bind to the operator region. I'll show you what that looks like in a second. Basically blocks RNA polymerase from transcribing the gene. And so when this gets induced, how does that happen? Well, think of it like this. If lactose is present in the environment, the bacterium is taking it up. It's like, hey, this is a good time to use this. Let's switch these genes on. We need them now. And so lactose will bind to the repressor. It's actually an isomer called allolactose, which is a small proportion of the lactose that's out there. You just get the idea. If lactose is present, it's going to bind to the repressor. That will make the repressor come off. So what does that look like? Here we have our repressor gene that's a little bit upstream producing this protein. The protein here is binding to the operator region. So when no lactose is present, there it is blocking the way from RNA polymerase. No transcription is occurring. And so here's that scenario where lactose is present. And again, specifically allolactose is the thing that binds. So here's our repressor protein. You normally would just be sticking to this operator region, just getting in the way. But when allolactose is present, it will bind. And this causes the repressor to change its shape a little bit, and it will fall off of the operator region. And then that leaves the door open now for RNA polymerase to read past that region and then into the actual genes, and transcription can occur. Okay, so which of the following molecules, when taken up by the cell, binds to the repressor so the repressor can no longer bind to the operator? All right, so you may remember that it was lactose or allolactose, but specifically that thing is called the inducer. And so that was the answer here. All right, let's look at positive control now. And guess what? We can actually use the same operon as being under the control of positive influence here as well. It's not just being repressed, but there is a positive control necessary in the lac operon too. And the scenario we can set up here is the situation where the cell is an environment where both lactose and, say, another carbon source, say glucose, are present. And given that situation, it is more efficient to use glucose as a carbon source. And if we look at the structure here, glucose, think back to cell respiration. Where are we starting? Glycolysis with glucose. With lactose, you actually have to split lactose first, so there's an energy cost. And then you can convert it to glucose and feed that into glycolysis. So it takes an extra step. So with both of these things present, it's preferable to use glucose, more efficient from an evolutionary perspective and an energetic perspective. So let's go through a couple of different scenarios now. Low lactose scenario, but glucose is high. And that might be in your culture medium. You're feeding your bacteria lots of glucose, great. So what's happening to the lac operon? Well, nothing, right? The repressor is sitting there because there's no lactose, and it's not being transcribed. There is a bit of a added detail here saying infrequent transcription. And this is just kind of highlighting the idea that this repression isn't perfect. And so sometimes RNA polymerase is going to squeak by, but it's not a significant amount. All right, so we've seen this already. What happens if both things are present? Preferably, E. coli is going to use glucose. That's cheaper to use. And so, lactose is here, so allolactose binds, it's the inducer, off comes the repressor. RNA polymerase can move through the operator now and transcribe these genes, but it still doesn't do this very efficiently. So it's weakly transcribed when glucose is high because there needs to be this other effect. We've taken the brakes off the car, but you need to give it some gas now. That's what happens when we have low glucose. So here's now the scenario. It's like, okay, we don't have glucose. Let's use the lactose. Lactose is present. Off comes our repressor. Yes, we know all about that. And then here's that positive control. This cyclic AMP receptor protein, or CRP, it's the gas. It needs to be present to get frequent transcription from this operon. And so really, this operon is under both positive and negative control. And to get frequent transcription, you need to have the removal of the negative control, we see here, and the addition of this positive control, take off the brakes and apply the gas. So how does this glucose level influence this CRP protein? Well, there's a few steps involved, and we can go through each scenario. So when glucose is high, it will bind to this adenyl cyclase enzyme. We've seen this before when we talked about cell signaling. So this is actually an enzyme we find in both prokaryotes and eukaryotes. Here we're talking just about prokaryotes. But when this is inactive, its job normally would be to make cyclic AMP. And so when it's inactive, you don't have very much cyclic AMP. cyclic AMP needs to bind to this CRP. It's called the cyclic AMP receptor protein. So it has to basically stick to it here. And so without it, you won't get the gas on the operon here. It will not have the positive control. So when glucose is low, you do have active adenyl cyclase, cranks out tons of cyclic AMP. Now there's plenty of that to bind to the CRP, and you have that positive control. The last bullet here is a little side note. Cyclic AMP is not just important for influencing the regulation of the lactose operon, but actually several other operons and over 100 genes in E. coli. Okay, so now you've learned a little bit about that lac operon. So what environment would lead to the most transcription of it? Okay, so to get the most frequent transcription, that's the last example we talked about. You need the positive control, so that means you need a lot of cyclic AMP to make this CRP bind, and you need the repressor to be off of that operator region. Okay, so here's another scenario. Suppose an experimenter becomes proficient with some genetic engineering to move some DNA sequences around. And what she does is she moves the promoter of the lac operon to a spot between the LAC-Z and LAC-Y genes. Those code for the beta-galactosidase and permease proteins. What will happen? All right, so yeah, given the choices here, what will happen is you've skipped over the beta-galactosidase, and the promoter could still be used to produce Y and A here, but you no longer have a promoter that's in front of beta-galactosidase, so you will not be able to produce that gene. And you could imagine all kinds of scenarios like this. If we, what if we deleted the operator from this? That would say take out the negative control, the ability for the repressor to bind there if we took that out. Or we took out this cap binding site or CRP binding site, sometimes also called the cap binding site. Then you wouldn't have the ability for that positive control factor to bind. So you can imagine all kinds of scenarios of moving things around and what the consequence of those would be. All right, so those are prokaryotic operons and how they're regulated in general. And the next session, we'll talk about eukaryotes and regulation of their genes.